Welcome. Today, we're diving into the work of Dr. S.K. Singh, focusing on the bioanalytical evaluation of targeted drug delivery systems. We'll explore how these systems are designed and tested to improve precision in medicine, making treatments more effective and safer. Let's get started. Let's start by looking at what analytical method transfer, or AMT, really means. It's a formal process that shows an analytical procedure works just as well when moved from one lab to another. The goal is clear: to make sure results stay reliable, no matter where the testing happens. This supports everything from site transfers and proving lab capability to keeping product quality high and meeting regulatory demands. Typically, the method moves from a sending lab to a receiving lab, and the key is achieving equivalent performance between them. Let's take a closer look at the regulatory framework guiding our work. We rely on key ICH guidelines, like Q2 for validating analytical procedures, Q14 for developing them, Q9 for managing quality risks, and Q10 for maintaining a pharmaceutical quality system. On top of that, USP sets specific requirements, such as those in Chapter 1224. The FDA adds its expectations, focusing on data integrity, process validation, and CGMP compliance. Finally, the WHO provides technical guidance to ensure method transfers meet good manufacturing practices. Together, these form the backbone of our quality approach. When we talk about transferring analytical methods, there are a few key types to keep in mind. First, comparative testing, where both labs analyze the exact same samples to see how results match up. Then, co-validation. This involves partially or fully revalidating the method at the new lab. Method verification focuses on confirming critical performance aspects. Revalidation happens when major changes to the method occur. And it's important to know that regulatory bodies usually prefer comparative testing as the go-to approach. When we talk about transferring a method from one lab to another, there are some key prerequisites to keep in mind. On the transferer side, the lab needs a validated method, an approved procedure, and solid historical data showing how the method performs. On the transferee side, the lab must have qualified analysts, the right instruments, a controlled environment, and approved standard operating procedures. Plus, the process relies on essential documents, like the transfer protocol, validation report, method SOP, and a risk assessment. All these pieces ensure a smooth, reliable transfer. Let's talk about risk assessment tools, the methods we use to spot what could go wrong. FMEA, or Failure Modes and Effects Analysis, helps us break down potential failures step by step. The fishbone diagram digs into root causes, showing how different factors connect. The risk ranking matrix then prioritizes these risks, so we know where to focus. Here, we see key variables affecting analytical performance, like instrument differences and sample handling, both rated high risk. Understanding these critical factors is essential when transferring methods successfully. Now let's talk about experimental design, the backbone of any solid study. We consider how many analysts will be involved, the number of runs, and replicates to ensure reliability. The method transfer protocol defines the scope and which methods are included. Responsibilities are clearly divided between the transferer and transferee labs, with QA overseeing the process. Materials like standards, samples, and reagents are specified. Finally, acceptance criteria are set up front. They're predefined and backed by scientific reasoning. This structure keeps everything on track. Now let's look at how we designed our comparative testing study. We tested 3 types of samples: assay samples, impurity samples, and dissolution samples. To ensure reliability, we worked with 6 individual preparations, had 2 different analysts involved, and repeated the tests across multiple days when needed. Our main focus was on comparing key parameters: assay, impurities, precision, and accuracy. This setup helps us build confidence in the consistency and quality of the results. Here, we see a side-by-side comparison of assay results from 2 different labs, the transferer and the transferee. Each replicate shows how close their measurements are to the expected label claim, hovering right around 100%. This table highlights the consistency between labs, confirming that the assay performs reliably no matter where it's run. It's a crucial step in demonstrating that the method transfers successfully and maintains accuracy across different settings. Now, let's focus on precision evaluation. This is about measuring how accurately our system performs, how close it gets to the target outcome without unnecessary errors. Precision tells us the quality of what we capture, not just the quantity. It's a key metric for understanding effectiveness, especially when false positives can be costly. By honing in on precision, we ensure our results are reliable and meaningful, setting a strong foundation for the next steps in our process. Let's look at how we evaluate success statistically. Here, the transferer and transferee means are nearly identical, 99.88% and 99.92%. The difference is just 0.04%, well within the acceptance limit of plus or minus 2%. This tells us the method transfer is successful. To reach this conclusion, we use several tools: mean difference, percent recovery, t-tests, confidence intervals, and equivalence testing. Each one helps confirm that the methods truly match. Acceptance criteria are the clear, agreed-upon conditions that a project or task must meet to be considered complete. They set the bar for success, making sure everyone knows what done really means. Without them, it's easy to miss the mark or end up with misunderstandings. So, acceptance criteria keep us aligned, focused, and moving forward with confidence. When transferring methods, we often face a handful of common challenges. Instruments vary, different HPLC systems, and detector sensitivities don't always match. Columns bring their own quirks, with lot-to-lot differences and aging effects that impact performance. Analysts add variability too, especially in how samples are prepared. Then there are environmental factors. Temperature and humidity can quietly shift results. On top of all this, data integrity risks loom, like uncontrolled spreadsheets and missing audit trails. These are the hurdles we need to navigate carefully. This slide outlines the key elements of our method transfer report. First, we confirm that the transfer objective has been met. Then, we present all experimental data, raw numbers, chromatograms, and calculations. Next comes the statistical evaluation, where we compare results to ensure consistency. Any deviations are thoroughly investigated, with their impact assessed. Finally, the conclusion states whether the method was successfully transferred or if corrective action is needed. The report closes with QA approval, authorizing the final GMP release of the transfer activity. Let's wrap up with the key conclusions. Analytical method transfer isn't optional. It's a regulatory must whenever labs or sites change. We rely on guidelines like USB 1224, ICH Q2, Q14, and Q9 to shape our approach. Risk assessment drives how we plan and design the transfer. Comparative testing is still the go-to method, backed by solid statistics and clear acceptance criteria. When done right, method transfer guarantees reliable data, high product quality, compliance, and smooth business operations. That brings us to the end of our presentation. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you.